Alrighty, guys. It's a quick presentation on viruses. I did do this presentation up a few years ago, and I did modify it slightly today with some stuff with COVID-19. But when I first did this, the real big scare was Ebola. And as you know, in about was it, about maybe 4 or 5 years ago, we had a big Ebola outbreak in West Africa, and then that actually got out to a couple other countries and things, and we were worried about it here. We even brought some people to the United States, Americans who were infected, and people were freaking out. Oh my God, we got Ebola! Something's going to happen. Ebola is really hard to get. Okay, it's a hard virus to get. The only way you can get it is by physically interacting with somebody who has body fluids leaking out of their body. That's where the virus is. Now, the reason why it was so prevalent in Africa has to deal a little bit with the funeral rites that they did, where somebody died, they would take the body, all of the close family members would get together, and they would help to wash and prepare the body and things of that nature. Okay, and that's one of the main reasons why Ebola spread so rapidly through Africa. And of course, when those cultural norms got interrupted because the medical people said you cannot do this, and they would come in and take the body, that of course, as you could imagine, really caused an uproar amongst people who were so used to being very intimate with the dead, so to speak, in terms of taking care of the body and go through the customized rituals that we used to do too, but we don't do that anymore because we send them off to the funeral home. Okay, well, they're a little bit different nowadays. Tetanus. This is what destroyed Napoleon's army. Okay, tetanus will it's a virus that will rest in soil, especially where horses and livestock have been. And once you get it, you start going through muscle spasms, and these muscle spasms are so severe you can actually snap your back, break your back. And that's kind of what's happening with this baby right here. It's going into this muscle spasm, and of course that is not going to live very long. And hepatitis, which is a viral disease that affects your liver, and of course, this person is also not going to survive when hepatitis is that far gone. Okay. So, anyways, just want to kind of quickly look at eukaryotic cells are pretty large compared to a bacterial cell, and viruses are even smaller compared to the bacterial cell and the eukaryote. So viruses are really super tiny, okay, and they're really super simple. And again, basically what it is, and we talked about this when we talked about the structure, when they tried to figure out what is the hereditary material in a cell. We looked at a couple of experiments where they were using bacteriophages. Why? Because bacteriophages are made up of basically a nucleic acid and a protein, and those were the two leading candidates for the molecule of heredity at the time. Okay, they are not cells, they are infectious particles composed of nucleic acid and a protein coat. Some viruses, as you guys learned when you went through that click and learn, also have a membrane coat around them, which is derived from the host cell's cell, basically. They're tiny, all right? They're really tiny. Different structures. We got the protein, the nucleic acid on the inside, and as you guys know, it can either be RNA or DNA. And not only can it be RNA or DNA, it can be single-stranded RNA, it can be double-stranded RNA, it can be single-stranded DNA, it can be positive sense RNA, or it could be negative sense RNA. Okay, so there's a lot of different things, different types of nucleic material that can act as the hereditary info in a particular viruses. Okay, um, but they are small, and as I said earlier, some viruses do have an envelope. And that envelope does come from the host cell's membrane. And very typical of many viruses are these various types of glycoproteins. Remember, glyco means sugar, protein, of course, you know what it is. So these are a sugar protein molecule that plays a role in getting, or I should say, gaining entry or access to the host cell. They're kind of like the keys, if you will, whereas the lock is going to be the receptor proteins on the host cell. Now, does the host cell have receptor proteins that were designed for the virus? Heck no. Those receptor proteins are meant for other types of activity, cell communication within that particular cell. It's just through the process of natural selection that viruses have formulated proteins that do fit those. Okay. All right. So once again, you went through. You learned about bacteriophages, which only infect bacteria. They're really small. Okay, as you can imagine. But a couple of things right up here. Now, like I say, viruses can reproduce only within a host cell. They're obligate intracellular parasites. Obligate intracellular parasites. They cannot reproduce on their own. And one of the reasons why most biologists will say they are non living, it's still up for debate in many circles. Okay? So, like I said, an isolated virus is unable to reproduce or do anything else except infect an appropriate host.
Wait, not living. How do they like come back to you?
They use the host cell to do all their replication.
So they
So,
stay there until like next year.
I mean, yeah, a virus by itself, if not one that is covered with a host cell membrane, but a virus that is just a protein and nucleic acid, those can actually be crystallized and then be uncrystallized and still infect a host. They may be viable for tens of thousands of years, which is happening. We see this up in areas like in the permafrost. You all know what permafrost is? Okay, continuously frozen ground up in the Arctic Circle. As that permafrost is now thawing due to global warming, they're starting to detect novel viruses and novel bacteria coming out of it. Okay, novel meaning new to science. All right, most viruses are new to science because we only have identified so many of them. But if you go swimming in the ocean and swallow some water, you probably swallowed several tens of thousands of viruses of probably 1,000 different species. Okay, it's just that your body is not a host for those viruses, so they don't affect you at all. Okay, and again, a isolated virus is merely a packaged set of genes in transit from one host cell to another. Kind of like that. But again, each virus infects or parasitizes a limited host range. And you guys, when you went through that click and learn, you saw that. What was the only one that infected plants?
Uh, T-
TMV, tobacco mosaic virus. What's cool about that virus is that some um, botanists, molecular botanists, have taken that virus and have utilized it to actually change the color variations in leaves of certain types of flowers, like tulips. So if you ever see tulips that have kind of you know a two-colored pattern in the petal, like a yellow and an orange, maybe it's kind of like looks almost like a psychedelic type drawing. That's a result of implanting viruses into the plant itself. Okay. Viruses identify host cells by a lock and key fit between proteins on the virus and receptor molecules on the host cell's surfaces. Some viruses, such as rabies, do have a broad host range and can infect several species. Rabies can infect humans, it can infect dogs, it can infect raccoons, skunks, marmots, bats, many, many different critters. Okay? And what did you learn about rabies? Did you learn anybody learn anything about rabies when they went through that click and learn? What happens once you start showing symptoms of rabies? What's that? It's fatal. Once you start, you can get bit by a rabid animal, and as long as you get taken care of and you get a rabies vaccine quick enough, you are okay. But as soon as you start foaming at the mouth, okay, forget it, you're dead. All right, so I've been vaccinated against rabies a couple of times because I went to Africa because I was working with animals that could potentially bite me all the time. All right, and again, not only are many of these viruses species specific, they're also tissue specific. Think of the common cold. Okay, common cold might only infect a limited number of organisms, like say common cold can infect me. It could probably also infect several of the great ape species. Why? Because we're closely related. So if you ever go gorilla trekking in Rwanda or maybe in the Congo or maybe even in Uganda, if you have a cold, they won't let you go. Okay, because you can still transmit that virus to them. Alrighty, so we went through and talked about you did the quick and learns on viral infection, and in general, the virus gets in the cell, okay, releases its nucleic acid. That nucleic acid is going to do a couple of different things. It's going to replicate itself. It's also going to make messenger RNA to make all the proteins that it needs to do, and all of this is utilizing the host cell's machinery. The virus doesn't have it. The virus does not have ribosomes. The virus doesn't have Golgi apparatus. Doesn't have any of that. But it will then utilize the host cell to produce new viruses, and that's your basic viral infection. Okay, looking at bacteriophages, the ones that infect bacteria. Okay, we can look at two slightly different versions of those replication cycles. One is called the lytic cycle, and lytic refers to lysis. Remember, lysis means to break apart, and this is generally of the more virulent the more virulent viruses. Virulence refers to their lethality. The more virulent something is, the sicker you'll get, the quicker you'll die, so on and so on and so on. Okay, so first of all, here we have a T4 bacteriophage that has DNA as a genetic material, injects its DNA into the cell, and immediately that DNA starts to make more copies of DNA. It starts to make protein. It starts to use the RNA polymerases inside there to transcribe and translate genes to make the viral proteins. And very quickly, those viruses are built and they cause the cell to basically blow up to lyse, releasing a lot more viruses. The enzyme that we use in our lab, the restriction endonuclease, the DNA scissors, okay? These DNA scissors, these restriction endonucleases come from bacteria because bacteria are trying to prevent themselves from being destroyed by these bacteria, by these viruses. Okay, so the restriction endonuclease of a bacterium is a type of immune system that they have evolved to protect themselves from bacteriophage attack. Okay, and we have now utilized or learned how to utilize those restriction endonucleases for molecular biology techniques, such as you guys did. All right. The lysogenic cycle is another cycle that you see with bacteriophages. It's what we call temperate. And what zone do we live in ecologically? The temperate zone. What's the temperate? It's neither too hot, it's not too cold, it's just right. Okay? So a temperate zone it implies something milder. Okay? It's not as mild really. So what happens with this temperate cycle phages is that they inject their DNA into the cell, okay, and then that DNA gets incorporated into the host cell's genome. It becomes part of their genome. Once it does that, it's known as a prophage. And that prophage will stay in that host cell. And every time that host cell replicates, it's also going to replicate the viral genome. And at some point during this cell cycle, it may receive a signal, whether it's environmental or whatever, it depends, is that prophage will now come out and will start to replicate its bacterial DNA or RNA and also start to replicate the proteins of that virus and start to make more viruses and eventually cause lysis of that cell and the release of those viruses into the environment. So the lysogenic cycle is where the genome of the virus becomes incorporated into the host cell's genome, but it can kick off a lytic cycle. Okay? Same thing that happens with HIV infection. When you get infected, or if you get infected by HIV, that viral genome gets incorporated into your helper T cells. Okay? So here's a question. If the viral genome gets incorporated into your helper T cells, can you now pass that on to your offspring? Yes or no? I see yes. No. The only way you could pass a viral disorder onto your offspring, you know, in utero, well, I should say genetically, is if that mutation or that virus infected what? Would have to infect what kind of cell? A gamete cell, either your sperm cell or your egg cell. Okay? Yes, a pregnant mother could pass on a virus to her offspring. Okay? In the womb. That's why they're looking at right now that the women who are pregnant and get COVID now are having a lot. What was it? Like a 40% greater chance of dying and a 50% greater chance of ending up in the hospital. Okay, but I don't think their child will have COVID. So, but things like HIV that can be passed on through breast milk. Okay, so if a woman's HIV positive, most likely she will be asked to bottle feed her child. Okay, because otherwise that potentially pass that on to the child. So here's an example. Here's the lysogenic cycle. The viral bacteriophage comes in, injects its DNA. Viral genome gets incorporated into the host cell genome. It's now known as a prophage. Then it gets a signal, starts moving into the lytic cycle, starts to assemble new viruses, and eventually that cell lyses, releasing those viruses into the environment. All right. So if that virus now has a protein envelope or now has a membrane envelope, then once again, that virus is going to fuse with the host cell, release the inner capsid, and then the viral genome will again copy its genome, make messenger RNA molecules, self assemble, and the virus then bugs out. What's that?
Is the lytic and lysogenic just affecting bacteria?
Yes, they are. The lytic and lysogenic cycles are pretty much just with bacteria. However, we have a very similar type of situation with eukaryotic cells. Something like herpes. Okay? Herpes virus gets incorporated, the genome gets incorporated into your nerve cell endings. And it'll sit there when well, the nerve cells don't really replicate, but it will sit there until certain environmental clues trigger it, and then you might get an outbreak like a cold sore or something of that nature. Okay? Or chicken pox, very similar. It sits in your nerve endings, and then it can erupt later on in life, and you end up with shingles. You might have heard of shingles. Okay, somebody having shingles. Now, if your parents are my age or close to my age, they should probably get the shingles vaccine if they have not yet gotten it yet. Okay, because shingles is extremely painful. Alrighty, you definitely don't want to get it. So here's just another animation, kind of another one where we see the envelope virus coming in, docking in with surface proteins. That's going to let the virus in via a form of endocytosis. The capsid is released, the proteins are degraded, releasing the viral genome, and then we start the whole process of making more nucleic acids, making the viral proteins, and then budding is what the release of that virus is going to be called. Alrighty, so there's lots of different types of viruses, especially different types of herpes, simplex 1, simplex 2. Chickenpox is also a version of the herpes. Epstein-Barr, you might have heard of that before. That's that virus that kind of makes you really tired. A good buddy of mine got Epstein-Barr. He was in college, he's my age, in college, and he's had it all his life, and it's really kind of affected him pretty badly. So he really had a hard time keeping a job and things of that nature because he just kept on like falling asleep and all sorts of stuff. So lots of different variations on the herpes virus. Okay, you learned that some of the viruses had RNA versus DNA. Okay, some of those RNA viruses have what's called a plus sense RNA. The plus sense RNA acts as messenger RNA. Okay, others have a negative sense RNA, and then that RNA has to be acts as a template for making the messenger RNA, which then goes ahead and makes the proteins necessary for the virus. I think COVID was a negative sense virus, so on and so on. HIV, horrible disease, incredible virus that in terms that enabled us to learn so much about basic cell biology, our immune systems, and also about virology. It was amazing because the amount of money that got poured into the HIV research. I was 18 years old in 1980 when people started dying from HIV. And I don't know if you guys know anything about the whole history of HIV in the United States. What population was hit the hardest early on in this what's that? Yeah, homosexual population in the United States. Part of that had to do with some of the practices that were going on, where there were these bathhouses, okay, where homosexual men would congregate and just have anonymous sex all the time, and those viruses were being passed all over the place. Okay, sexually transmitted diseases have a tendency to get transmitted very quickly. Now, what we know is that now that HIV probably made it to the United States through travelers and certain populations and whatnot. Um, the origin of HIV is in West Africa, around the Congolese Cameroon border. That was found out by a researcher here at UAB, Dr. Beatrice Hahn. Tracked it down. Its origin is it comes most likely from a combination of sooty mangabees, which is a type of primate, and chimpanzees. Um, it, you think, according to what Dr. Hahn says, is that the origin is in sooty mangabees. Chimpanzees ate sooty mangabees, got the virus into chimps, and then that virus became temperate in the chimps. So we believe that this virus probably first originated close to 1 million years ago, became innocuous, meaning it didn't cause any type of sickness in chimps after probably several tens of thousands of years of them coexisting together. And then humans, through the process of hunting chimps for